Yeah, so we are coming up with the next session. So you can take the slide, you can check the slide with respiration 2, part 2. Okay. Relax. Can you check here? This one. This one. It is passive process as diaphragm and intercostal muscle relax, and in inspiratory phase, the muscle contract. Okay. This is done. नेक्स्ट है द रेट एंड डेप्थ ऑफ ब्रीदिंग. सो व्हाट इज़ द नॉर्मल रेट. अब आपसे पूछेंगे क्या नॉर्मल रेट है ब्रीदिंग का. कैसे हम पता करेंगे कि आपका जो ब्रीदिंग रेट है वो स्लो है या आपको इश्यूज़ हैं ब्रीदिंग में. तो उसका एक नॉर्मल रेट हमेशा हर चीज़ का एक नॉर्मल रेट होता है. सो नॉर्मल रेट ऑफ ब्रीदिंग इज़ 14 to 18 पर मिनट. तो 14 से 18 बार आप सांस ले चुके होते हैं. रेट डिपेंड्स ऑन द फिजिकल एक्टिविटी. ऑब्वियसली कितना आप रेट ले रहे हैं दैट डिपेंड्स अपॉन योर फिजिकल एक्टिविटी वेदर इट्स एक्टिव और नॉट. हेल्थ स्टेटस किस तरह का हेल्थ स्टेटस है. इमोशनल स्टेट रिलैक्स स्टेट ऑफ माइंड में है या स्ट्रेस्ड है. In normal breathing, the intercostal and diaphragm muscles are involved. In deep muscles, accessory muscle of respiration are involved that include sternomastoid, the pectoralis major and platysma. And contraction of these muscles increases the capacity of thoracic cavity. And during this, there is a volume of air in lungs called pulmonary volume. Next is what is pulmonary volume. The volume of gases in the lung under different condition of respiration is called pulmonary volume. मतलब lungs के अंदर gases की volume को आप बोलेंगे pulmonary volume. The volume of gases in the lungs under different conditions of respiration. पहला है टाइडल वॉल्यूम. कितने तरह की है. बहुत तरह की है. सो टाइडल वॉल्यूम होता है. व्हाट इज टाइडल वॉल्यूम. द ड्यूरिंग क्वाइट ब्रीदिंग द अमाउंट ऑफ एयर टेकन इन और गिवन आउट. कितना हमने नॉर्मल ब्रीदिंग में क्वाइट ब्रीदिंग में हमने कितना एयर ले लिया या छोड़ दिया उसे हम टाइडल वॉल्यूम कहते हैं. जो वॉल्यूम अप्रॉक्सीमेटली 500 ml होता है. इंस्पिरेटरी रिजर्व वॉल्यूम. रिजर्व मतलब कोई स्टोरेज किया गया है. रिजर्व का आपको मालूम होगा ना दैट द वर्ड मीनिंग. रिजर्व मीन्स स्टोरेज किया हुआ. द एयर टेकन इन बाय फोर्सफुल इंस्पिरेशन ऑफ अ टाइडल वॉल्यूम. एकदम से फोर्सफुली आपने कोई वॉल्यूम लिया तो उसको हम बोलते हैं इंस्पिरेटरी रिजर्व वॉल्यूम. तो कितना हो गया 2000 to 3300 ml. इंस्पिरेटरी कैपेसिटी. व्हाट इज कैपेसिटी. किसी भी चीज़ की कैपेसिटी या एबिलिटी. इट इज़ द टाइडल वॉल्यूम and the volume of air taken during maximum inspiratory effort is called inspiratory capacity. कितना एफर्ट लगा के maximum एफर्ट लगा के जो एयर या वॉल्यूम हमने इकट्ठा किया तो उसको कहते हैं इंस्पिरेटरी कैपेसिटी. उतनी ही कैपेसिटी थी मेरी कि मैं उतना ले पाया मैक्सिमम एफर्ट लगा के. ठीक है. सो इट इज़ कॉल्ड इंस्पिरेटरी कैपेसिटी एंड इट इज़ 3500 ml. एक्सपिरेटरी रिजर्व वॉल्यूम. इट इज़ द वॉल्यूम ऑफ एयर एक्सपेल्ड आउट बाय फोर्स एक्सपिरेशन. कितना आप बाहर छोड़ सकते हैं फोर्स लगा के आफ्टर नॉर्मल एक्सपिरेशन. मैंने एक नॉर्मल एक्सपिरेशन किया और उसके बाद मैंने फोर्सफुली सारी एयर को बाहर निकाल दिया तो उसको बोलते हैं एक्सपायरेटरी रिजर्व वॉल्यूम. Residual volume. What is residual volume? It is a volume of air that remain in the lung after maximal expiration. So when we do maximum expiration, the volume remain in the lung. So it is not that air never be 0%. Okay, in lungs. So it is maximum expiration and the volume left in the lung is called residual volume. Then functional residual capacity. It is the volume of air remaining in the lungs after normal expiration. It varies for 2500 ml to 3000 ml. It is sum of residual volume expiratory reserve volume. Then total lung volume. It is the volume of air remaining in the lungs after maximum inspiration. It is the sum of vital capacity and residual volume. It is about 5000 to 6000 ml. Vital capacity. It is the amount of air looked up in the respiratory passage. The air in these spaces are not available for exchange, and it is called anatomical dead space. The volume is about 150 ml. Interchange of gases between the lungs. Interchange of gases occurs between the blood in the capillary while surround the alveoli and the air in the alveoli. The alveoli are the sac-like structure unit for lungs where oxygen is exchanged for carbon dioxide. Composition of air क्या क्या है आपके air में? 21% oxygen, 0.04% carbon dioxide, nitrogen is 78%, and other gases. And what is partial pressure? Partial means part of pressure. The proportion of total pressure provided by each gas is called partial pressure. If the atmospheric pressure at sea level is 760 mmHg, then partial pressure of oxygen PO2 कैसे निकालेंगे? Then partial pressure will be PO2 minus 21 upon 100 multiplied 760 is equal to 160 mmHg. So partial pressure of oxygen is 160 mmHg. The same formula applies for carbon dioxide. The partial pressure of carbon dioxide is PCO2 is equal to 0.04 because 0.04 is present in air, right? So you can check from here. Can you check 0.04? कहाँ से आए 0.04? So 0.04 percentage is already there in the air. So we just need to see 0.04 upon 100 multiply 760 is equal to 0.3 mmHg. What is transport of oxygen? Oxygen is carried by two ways, by plasma or by RBC. One is by plasma, the other one is by red blood. Plasma at a tension of 100 mmHg, 0.3 ml oxygen is dissolved in every 100 ml of blood. By red blood cells, oxygen combines with Hb and it transported around the body, and it releases oxygen to cells that need to form oxyhemoglobin. Hemoglobin is a protein made up of 4 subunits, each of which contain a heme moiety attached to polypeptide chain. Each of 4 iron atoms, iron remain in the ferrous form, resulting in combination with oxygen is called oxygenation process. Hb4 plus O2 is equal to Hb4O2. Hb4O2 plus O2. Again, the additions of oxygen, it will give you Hb4O4 and so on. Then again Hb4O4 plus O2 is Hb4O6. The tension of oxygen in arterial blood is about 90 to 100 mmHg and the oxygen tension is less than 40 mmHg. When arterial blood passes through the tissues, it carries oxyhemoglobin. As the oxygen tension in the tissue is much lower, the oxygen tension in arterial blood falls. The equilibrium of oxyhemoglobin at various partial pressure can be studied through oxygen dissociation curve. So oxygen dissociation curve, what is that? The curve obtained when different partial pressure of oxygens are plotted against the amount of oxyhemoglobin formed or dissociated. तो ऑक्सीहीमोग्लोबिन या तो बनेगा या छूटेगा, क्योंकि ऑक्सीजन जो है हीमोग्लोबिन के साथ जुड़ जाता है, ठीक है। तो ऑक्सीजन या तो बनेगा या टूटेगा। तो कर्व जब हम बनाएंगे, जब पार्शियल प्रेशर, क्योंकि जब ऑक्सीजन जुड़ेगा या टूटेगा तो पार्शियल प्रेशर ऑफ ऑक्सीजन भी डिफरेंट होगा हर समय पर। तो हमने क्या करना है, डिफरेंट जो हमने नोट डाउन किए हैं पार्शियल प्रेशर ऑफ ऑक्सीजन, उनको प्लॉट किया जाएगा अमाउंट ऑफ ऑक्सीहीमोग्लोबिन से, एंड द सिग्मोइड कर्व इज कॉल्ड ऑक्सीजन डिसोसिएटेड कर्व। द कर्व इज अ ग्राफ दैट शोज परसेंट saturation of Hb at various partial pressure of oxygen. This curve shows the equilibrium of oxyhemoglobin and non-bonded hemoglobin at various partial pressure. At high partial pressure, Hb binds to oxygen to form oxyhemoglobin. On saturation, erythrocytes are in the form of oxyhemoglobin. As deprived erythrocytes PO2 decreases, oxyhemoglobin releases the oxygen to form Hb. And how transport of carbon dioxide takes place? Carbon dioxide is carried in blood in 3 ways: in solution, in combination with protein, and as bicarbonate. In solution, the carbon dioxide is more soluble than oxygen. Its amount is proportional to tension. At tension of 40 mmHg, 3 ml carbon dioxide dissolves in every 100 ml of blood. Carbon dioxide in solution forms carbonic acid that ionizes at blood pH into hydrogen and bicarbonate ions. CO2 plus H2O, H2CO3 gives H plus HCO3. In combination with protein, carbon dioxide forms neutral carbamino compound with Hb. It combines with Hb in red blood cells. The oxygen combines with iron radical. The carbon dioxide combines with amine group of protein, forming carbamino group with fully reduced Hb. 8 ml of carbon dioxide is carried as carbamino by 100 ml of blood. As bicarbonate, the maximum portion of carbon dioxide in the blood is the form of bicarbonate, sodium bicarbonate in the plasma, and potassium bicarbonate in RBCs. As the blood passes through capillaries, bicarbonate ion diffuses into plasma. So I think that this is easy language and you can understand. So as the blood passes through the capillaries, when the blood passes through the capillaries, the bicarbonate ions diffuses into the plasma and the protein and ions cannot cross the cell membrane. Sodium and potassium do not diffuse freely. Electrochemical neutrality is maintained by diffusion of chloride into red cells, and the phenomenon is called chloride shift. So it is very important to understand what is chloride shift, and you need to explain it also when you are writing about transport of carbon dioxide. So you have to write about in solution method, in combination with protein, as bicarbonate, and you have to mention chloride shift. It is easy. In arterial blood, 3 ml carbon dioxide are in sodium, 3 ml carbamino and 2 ml as bicarbonate, making a total 48 ml carbon dioxide in blood. So the blood leaves the lung and arrives at tissue carrying 48 ml carbon dioxide per 100 ml at tension of 40 mmHg. It leaves the tissue and arrives at the lung carrying 52 ml carbon dioxide per 100 ml of blood at 46 mmHg. The three forms of carbon dioxide are carried by the blood and they interact to form total carbon dioxide dissociation curve. So you can check the curves downstairs, down here. And the carbon dioxide dissociation curve is plotted as carbon dioxide 100 ml of blood against partial pressure. If the carbon dioxide tension increases, the total amount of carbon dioxide taken up blood increases at any given carbon dioxide taken up blood also. So carbon dioxide dissociation curve, x-axis and y-axis. The x-axis, the partial pressure of carbon dioxide in mmHg, and y-axis we are having content, carbon dioxide content, ml CO2, right? And this is, we are plotting that, and total carbon dioxide. So here you can check this one, total carbon dioxide. This is oxygenated blood here, this is oxygenated blood, okay? And carbamino, carbon dioxide is here, so it is going dissociating from the blood. So that is why it is called carbon dioxide dissociation curve. So here is the same thing. If you see, PO2 is in x-axis and oxyhemoglobin is here, and left shift decrease temperature, decrease this, and this is your carbon and this is your oxygen. This is here, okay, oxygen is there. So oxyhemoglobin, the saturation point, after saturation point it has to dissociate because the maximum limit is reached. So I hope you understood that, and please go through the previous video also, because I have explained it there as well. These are the notes, just take care of the notes. If you don't understand, please revert back in Telegram or YouTube as a comment so that we can check it out. And next time whenever we do this Zoom callings or any video calls for your clearance of doubts and all, so we can check it out, right? Thank you so much and hope you study well. Exams are exams will be anytime. Okay, so please study hard. Take care. Bye bye.